Welcome, everybody. I'm Paolo Cassano. Today is October 6, 2023. Welcome to the MGH brain-PBM clinic rounds. And it is my great pleasure today to welcome Dr. Liu and his postdoc, Luyao Tang, both from Fudan University. Dr. Liu is from the School of Information Science and Technology at Fudan University, Shanghai, China. And he will present with Luyao Tang about advantages of pulse-like PBM and its practice on enhancing cognition with tPBM. Welcome, Dr. Liu, and please take it away if you want to start sharing.
Yeah, thank you very much, Dr. Cassano. Thank you for your invitation to have a talk here. It's really my pleasure to meet so many international friends on tPBM society. Okay, I will share my screen. Can you see it?
Yeah, we do.
Okay.
Yeah.
Okay, thank you. Good night, everyone. This is now in China. It's midnight, so I hope I can awake enough to finish my presentation. Thank you for invitation, and also thank you for all the participants to join us. Today, my talk is about the advantage of a pulse-like PBM and its practice on enhancing cognition with tPBM. First, please allow me to do some more introduce about myself. I'm from Fudan University. This university located in Shanghai, China. And currently, I'm a full professor at the School of Information Science and Technology. I'm leading a PBM laboratory. And this group is roughly 20 people. And this is my contact information. Okay, now this is the contents of what I will talk today. When we talk about PBM, it's always associated with light therapy. But really, light therapy is not only in PBM. It also includes something else, just like high-power laser therapy. But really, this is quite different from the PBM. Since it destroys cells or tissues, even vaporizes it. So, and on the other hand, the PBM is biologically modulate cell and tissues by photons. Sometimes it's also called LLT. That means low-level light therapy or low-level laser therapy. Of course, today, my talk is only on PBM only. Let's have a memory about Niels. Roughly 100 years ago, he did...
Recording in progress.
Pardon? Okay, I continue it. Roughly 100 years ago, Niels cured smallpox by red light and lupus by ultraviolet light or blue light, and with big successful. So he won Nobel Prize in 1903. But then I will have a question that it's roughly 120 years after Niels, we can say PBM has some progress, but really it's too much. What's the reason about that? I think there are two limitations for this kind of light therapy. The first thing I will say is come from light sources. Since for PBM is featured with spectral selectivity, from these three fingers we can see that when the light acts on human visual corners or red cell, we can see different wavelengths, the response is different. We can see here have highest and here have highest. Again, when we look to the photosynthesis, that means the light acts on plant cell, just like PS1 or PS2 system. It also shows different wavelengths have different responses. Again, we look to another cell we call hello cell. And here we also see there are 4 peaks. So that means PBM is always featured with spectral selectivity. On the other hand, when we look to the light source available before LED, these are the 4 light sources available, just like incandescent lamp, and fluorescent lamp, and the sodium lamp, and the metal halide lamp. All these 4 lights are always have the spectrum like this. Of course, you can change the material or even change the voltage, slight change the spectrum, but really the change is not too much. I mean, the spectrum always like this. So in this way, there should be a problem. For PBM, need some special spectrum, but the light source only available this kind of spectrum. This is the first limitation. Of course, after LED development, roughly 20 years until now, since LED can have different kind of spectrum by changing the material inside. Furthermore, since the LED is very small in size, so in real application, you can mix up several LED or many LED to make the spectrum very flexible. So that means you can always find some optimal spectrum for PBM. This is the first limitation, but it can be mostly solved by LED. The second thing I will say that this is the contradiction of response and skin temperature. As here we see, this is well accepted by PBM's society. This is under the source model. It used to describe the PBM responses and light doses. In our research work, we also show the PBM response is not only related to light dose, but it's also related to irradiance. Here we have an example of the melanoma cell. Here we see with a lower irradiance, the inhibition rate is lower, and with higher, the inhibition also higher. So for this, when we look to the real application, for example, when we talk about melanoma, since this cell is deep inside under the skin, and there are many articles has the simulation about light penetration into the target cell. The light will have big attenuation. And this is also we in our group we measured for fat and lean, of course from pig, from pork. So when we talk about the melanoma, it's roughly 1 or 2 millimeters under the skin, and if you use blue light to treat melanoma, so the transmission is only roughly 2%. It's very, very low. If we use this data and this data, if you want to have the inhibition rate in this irradiance, that means the irradiance on the skin must be roughly 50 milliwatt per square centimeter. And this data is quite high to make the skin temperature is quite high. So that means if we want to have a higher response of PBM, we must, in many cases, we make the skin temperature quite high. So this is the second limitation. Okay, so I just mentioned the two limitations. The first limitation can be solved by LED's development. And second, again, can be use LED, but it must be used in pulse mode. I will talk about that a little later. Okay, now we can have a definition about the pulse light parameter, just like T on, T off, and the period, and frequency, and duty ratio, and peak irradiance. This data is very important, and average irradiance. For all these parameters to describe the pulse light PBM. Okay, now we want since we want to compare the PBM response efficiency of pulse light and continuous light. So here we compare this light A and light B and light C. Light A is the irradiant. Light A and light B, they are both continuous light, but the irradiance is only this one, and the light B, the irradiance 3 times of that. And here we have another light C. Light C is the pulse light, and frequency and duty ratio, and the times like this. Here it's easy to understand. The average irradiance of light A and light C are same. Here again, we use the finger I just mentioned about, but I rotate it in 90 degrees to make it very clear. Since here we see light A, the PBM response is roughly 6% or 6% roughly, and the light B is like this, is more than 10%. And now we want to ask, how about light C? Since it's even light B, the PBM response is higher than that, but since this one will cause the higher temperature in skin surface, so I think this, in many cases, this is not good enough, since even the response is high, but higher temperature make it use this in many cases. So that really we must check, how about this one? Since this one and this one, the temperature are exactly the same. If the PBM response of this one is higher, then it should be very good. Okay, now I have 3 examples about this comparison. The first thing again is the melanoma. We design 5 different light conditions. Just this is the continuous wave, and this is the pulse light, and pulse light with different frequency. All the 5 light conditions, they have same average irradiance, but the duty ratio are same. And here we can see the inhibition rate of this one is highest. Of course, is higher than continuous wave. This is the first example. Again, let's look to another one. And in our group, we also do research on cyanobacteria, and we use here we use oxygen evolution rate to as an indicator of PBM response efficiency. We set 3 different pulse light to test by this special machine to test instantaneous the data of a PBM response. And here we see the curve is very like an exponential function. And really, we use this formula to simulate the results with very high accuracy. So in this way, we think this PBM response is like a capacitor module. Then we establish an electronic circuit to simulate this photosynthesis process with very high accuracy. And so in this way, by this simulation, we can easily to understand this shadow area is just the increasement of pulse light PBM response compared to continuous wave light. This is the increasement. So this is the second example. Again, we have a look to another example, and this is about the human vision. Here we compare the perception enhancement ratio. That means that the human perception, the brightness ratio between the pulse light and the continuous wave light in some physical power. Here we see all the data are higher than 1, even 1.8, roughly 2 times in this data. Of course, all the data related to the duty ratio and the wavelengths. But anyway, we see all the data higher than 1. That means human perception brightness of pulse light is higher than continuous wave, even they are in some physical power. And this is a segment we use to do the research work. So, and all these data we have published in have 3 publications. Besides that, we also do something else, do the PBM pulse light on something else, just like we use argon to produce hydrogen, and we also use pulse light. We found some enhancement. And also if we use this bacteria, and also we also do pulse light on DPC, and this is for hair growing. And also this one, and also this one, and this one, and something else. We do all this for PBM on cells with quite high quick work. Since we thank you very much to the Shanghai Light Engine company, they supply us light computer, light incubator. And this light incubator has 8 separate chamber inside, and each chamber can control the light condition and temperature. So in this way, we can do the PBM on cell or some other tissues in 8 light conditions in same time. And this will speed up our research work greatly, and also ensure repeated accurate for 8 light conditions in some time. So let's conclude the question I just asked before. We compare light A and light B and light C. Then we know the PBM response of light B is higher than light A, but this one will cause high temperature on skin, so this is not good useful in many cases. But this one is also higher than this one. The PBM response higher than this one, and the temperature on the skin is exactly the same as this. So I think this should have many applications, especially. I think for since today we talked many of you from the tPBM society. I think for tPBM, the signals from the surface to the touched cell should be at least the 1 or 2 centimeter. So in this way, this is even important. So okay, but really we must test if the transcranial PBM should have pulse-light enhancement. I have talked about that, and this is very careful about from the tPBM Society. And this work is a part of my PhD student, the Tang Luyao's dissertation. So she will talk about that, then she will talk about that. OK， 汤洛瑶，你就是我退出了，你进去啊。
OK。您退出了吗？
我退出了。
OK。Can you see the screen?
We can. Thank you.
Okay. Hello everyone. My name is Luyao Tang, and now I'm going to give a talk on our recent findings regarding pulsed transcranial photobiomodulation on cognitive enhancement of healthy individuals. The content of this talk has been published on the journal Lasers in Medical Science, and I'm the first author of this article. Firstly, let me introduce transcranial photobiomodulation, abbreviation as tPBM, briefly. It is commonly conducted with red or near-infrared light irradiated on the scalp, as shown in this picture. Previous studies have proven its therapeutic uses for brain diseases in healthy individuals. Evidence from cognitive associated with beneficial emotional effects, enhanced executive functions, better attentional performance, improved rule-based learning, reinforced short-term memory, and so on. Meanwhile, tPBM's modulation on neural oscillations has also been reviewed. On high-order cortical functions, researchers started further exploration regarding the optimal parameters, including wavelengths, power density, fluence, operation mode, etc. And from the perspective of operation mode, most of previous studies focused on the continuous wave, namely CWT-PBM, while relatively few researchers focus on the pulse wave mode, and this aroused our interest in studying the differences between PW and CW transcranial light. We thought that PW transcranial light may bring about different results due to the renaissance effect with the natural brain oscillations, or higher PBM response brought by higher peak power density, as introduced by Professor Liu previously. And we conducted our studies through cognitive experiments and EEG signal analysis. Next, it comes to the materials and methods part. The light stimulation device was a self-made LED light source with 36 chips assembled on the aluminum substrate. The irradiation area was a 4 × 4 centimeter square on the right forehead. We recruited 56 healthy young adults, and they were randomly assigned to one of the following 4 treatments: sham, CW tPBM, PW tPBM with 40 Hz, and PW tPBM with 100 Hz. The lighting conditions are summarized in this table. The investigated wavelengths were 660 nanometers and 550 nanometers, which both match the action spectrum of CCO. The average power density was fixed at 250 milliwatt per centimeter square. Since the duty cycle was fixed as 50%, the peak power density of pulsed light was 500 milliwatt per centimeter square. We designed 3 psychophysical experiments to test the sleepiness, attentional performance, and short-term memory, respectively, to study the changes of subjects before and after receiving tPBM intervention. The first is Karolinska Sleepiness Scale, and abbreviation is KSS. KSS is a widely applied method for measuring sleepiness. It is a 9-point scale as shown in this table. Participants were asked to give their KSS scores by self-report before and after the tPBM procedure. We designed the psychomotor vigilance task as PVT experiment to detect the rapid response ability and concentration level of subjects. It was implemented by MATLAB and the Cycle Toolbox 3 Toolbox. One block of PVT consisted of 40 trials and takes about 2 minutes to finish it. During the task, left or right arrows or red dot randomly appeared on the screen. And the left arrows corresponding to press the F key button on the keyboard, and the right arrows was related to pressing the J key button on the keyboard. If a red dot appeared immediately after the arrow, the subjects were asked not to make any key press response, and the probability of the red dot appearing was set as 20%. Subjects were given a short practice session to familiar with the task. The post-treatment block of PVT trials was identical to the prior ones. Following the PVT was the delayed match to sample task as DMS, which tests the short-term memory as well as the reaction time. The task entailed weaving a 6x6 grid of yellow and purple squares with a randomly generated pattern for each of 20 trials. Firstly, the target pattern appeared in the screen for 5 seconds. When the observation time was over, the screen would be blank for 5 seconds as a stage of forgetting and recall. Then, a stimulus, either a match or non-match, would be presented on the screen. The match pattern was exactly the same to the previous target pattern, and the non-match contained 1 to 2 randomly switched squares, as shown in this figure, like this. So these two figures are similar but not identical. So the correct answer for this example trial would be non-match. The participant indicated whether the stimulus was the same as the previous target with the keyboard. The post-treatment block of 20 DMS trials was identical to the prior one, and the subjects took about 8 minutes to finish one task. When the KSS PVT DMS task were finished, we get subjects prepared for the EEG signal acquisition. And all EEG signal was collected by a 32-channel device, and the collection consisted of a 3-minute baseline period, an 8-minute exposure period, and a 15-minute rest time period. So the total duration was 26 minutes. The EEG data were pre-processed using the EEG lab toolbox on the MATLAB platform. It contains steps like removing noise period, filtering, sample rate adjustment, wrong ICA, and re-reference. The power spectrum analysis was conducted through the spectral function as implemented in EEG lab. It can return and plot the mean log spectrum of a set of data apples at all channels, and it's derived from the P-Welch function, which is responsible for calculating spectral power density as SPD. This figure presented an example of results derived from spectral function, and the absolute power of each frequency band were calculated as shown here. The flowchart of the whole experiment is shown in this picture. Each subject would only accept one of the 4 conditions as sham, CW, PW40, or PW100 for testing, and completed the cognitive experiments and the acquisition of the EEG signal successfully. With regard to the statistical analysis, for KSS, PVT, and DMS tests, Novelly distributed variables were analyzed with paired sample t-test using pre- and post-treatment data within each subject. Then, one-way ANOVA with Beforonis post-hoc tests was applied for between subjects comparison. For EEG data analysis, one-way repeated ANOVA test with multiple comparison for detecting baseline durational and rest time signals with Beforonis post-hoc test. And here comes to the Here comes to the results part. We can show that for KS score results, pre-post comparisons showed that in all groups of 660 nanometers, KS scores decrease, and the changes in CW and PWT PBM groups were significant. Under 850 nanometers, only PW lights decrease was significant. The decreasement of KS scores indicating that the subject self-evaluated sleepiness decreased. The between groups ANOVA test showed that there were no differences between the groups prior to intervention, and this meets our expectations. And after tPBM, KS score of CW and PW40 group of 660 nanometers as well as PW40 group of 850 nanometers was significantly lower than control group. As for the PVT experiment, I would like to define the rate correct scores, which equals to the number of correct responses divided by the sum of all action times. The higher the RCS scores, the better we think the cognitive performance. However, in this case, the RCS of PVT did not show any significant differences before and after tPBM or between groups, and this is this does not meet our expectations, and it's contradict to the previous findings. The most likely explanation for this is that our study contains a step of asking subjects not to press the keyboard immediately because he or she need to wait for the red dot to appear or not. And this asks them some more concentration level. So further, we counted the number of correct responses increased in PW groups of 660 nanometers and PW40 group of 850 nanometers significantly after tPBM. We think this can be a supportive clue that indicating that the subjects tended to be more concentrated on the PW light conditions. As for DMS experiment, we also use RCS scores and found that the RCS of PW group after tPBM increased significantly under 660 nanometers, and PW40 group of 850 nanometers showed similar trend. And this is a promising result, indicating that PWT-PBM achieved better intervention results compared with CW or sham control group. Like PVT, we counted the number of correct answers and finding that the increasement of correct answers was observed in PW40 and PW100 group of 660 nanometers after tPBM. As for the results of EEG signal analysis, there are two main findings. The first one is about the total power. We can tell that the total power of sham group decreased during and after the tPBM compared with baseline, but this was not observed in CW or PW tPBM group. And the second is about the gamma band power. We can tell that the 660 nanometers group here and here and here had a significant increasement of power in gamma band, while this was not observed in the sham group or CW groups. Since we think that the gamma band, as the gamma oscillations is related to high order cortical functions, we think the increasement of power in gamma band can be seen as a beneficial changes. And now I would like to comes to the discussion part. The first is about whether it is the frequency increment or penetration depth that are related to the advantages of pulsed light, pulsed wave tPBM. The data from our study showed that the difference between 40 Hz and 100 Hz was not very obvious in this study. And this may be attributed to the insufficient frequency parameters we set, like we didn't set 10 Hz or 1000 Hz, or that the frequency is not the key factor in PWT-PBM. And this remained to be investigated further in the near future. And as for the penetration depth, according to some researchers, light in PW mode has better penetration depth due to the higher peak power density in short pulses. And which might help us reaching the deeper structure of the brain. And supposing the light intensity at the cortex needs to reach a minimum threshold in order to generate a beneficial effect, then the on-time intensity of PW light may exceed the threshold, while the CW light's intensity is below the threshold. And because of the quench periods, means the T-off period following the T-on period, and this point has been introduced. I will summarize this briefly. And secondly, is about the tPBM's modulation effects on gamma oscillation. The average power within the gamma band of PW groups showed a significant increase during tPBM intervention, and we think it can be considered as beneficial changes. And Dr. Casano's teams also reported a significantly boosted gamma power in eye-closed EEG recordings, with the largest effects in the posterior regions. But this phenomenon also only occurred in CW light groups, not PW light or sham group. We think that the differences between Sparrow et al. And ours may be attributed to the parameters we set, as the tPBM's frequency is 10 Hz, and our frequency was 40 Hz and 100 Hz, but this remains to be discussed in the future. And the thirdly is about the PBM effect of pulse light on neurons. Literature has showed that retinal light irradiation on neurons can cause cellular changes on survivability, OS, MP, ATP levels, etc. And the comparison they carried out. So we designed investigations about the biological effects of pulsed red and near-infrared light on neural-like blastomal SHSY5Y cells by in vitro experiments. And we covered irradiation parameters, including wavelengths, dose, power density, frequency, and duty cycle, etc., and found that pulse light generated a distinct effect compared with continuous wave light on cellular responses, with measures of cell viability, OS, MP, and ATP. The intervention was finished by the incubator introduced above, and the wavelength was also 660 nanometer and 850 nanometers. The first experiment was about the intensity and dose. The intensity varying from 5 to 100 milliwatt per square centimeter. The irradiation time was 8 minutes, and the dose varying from 2.4 to 48 joule per centimeter square. And from the results, we can tell that the cell viability ATP and MP displayed a general trend of rising and then falling, corresponding to the biphasic dose response law. And except for the OS indicator, it showed an interesting bimodal at 20 and at 20 and 100 milliwatt per centimeter square. And this findings was consistent with the previous literature in 2008, but I forgot to put it here. But this is consistent with previous findings about the bimodal pattern. And we then set the average intensity as 10 milliwatt per centimeter square and change the frequency of pulsed light from 40 Hertz, 100 Hertz, and 1000 Hertz. And the duty cycle remained the same. And we found that the results of 40 Hertz PW mode light were significantly different from control group in 660 nanometer and 850 nanometer cell viability, ATP, MMP, and 850 nanometer ROS. The increasement or the differences was not very obvious, but it did show a significant difference. And the third one is about the experiment on duty cycle. The frequency was fixed at 40 Hertz, and the duty cycle varying from 10% to 50%. So the corresponding peak intensity was 100 to 20 milliwatt per centimeter square. And we found that the results of 20% and 10% duty cycle group were generally higher than control and 50 group. So we draw a preliminary conclusion that the pulsed PBM with a higher power, with higher peak power density can cause higher PBM response. So finally, I will make a summary about the two limitations of PBM is the unavailable of optimal light sources and contradiction of PBM response efficiency and skin temperature had been barriers for PBM applications previously in the past. And pulsed light with higher PBM efficiency compared to continuous light may bypass the contradiction of these two. And the findings of the study related to cognitive enhancement showed that PW transcranial light achieved better short-term memory enhancement effects compared with CWL light and could modulate high-frequency neural oscillations. In vitro study results show that the frequency and duty cycle had a distinct impact on neural cells. And for further details, please refer to our published article presented here. And that's all. Thank you for your attention. Thank you.
Thank you so much. I mean, this is a lot of data. Thank you, Luyao, and thank you, Muqing. So I think I'll open it to questions. And I might not be able to see everybody, so feel free to speak up if you have any questions, anything you want to ask him.
Did you actually get measurements of the surface temperature for the different wavelengths and some of the different power settings before the experiments, or did you just assume that these temperatures will get higher? You're muted, Professor Liu.
Yeah, thank you. Thank you for your question. I think the generally, if the irradiance on skin higher, the temperature will be higher. And we did measure the temperature in some cases. But since for real application, sometimes the light source will be far away from the skin. Sometimes if you use the wearable equipment, that means it's just touched to the skin. The two cases are quite different. For the second case, since the wearable tool is just touched to the skin and no air flow to lower the temperature in this time, generally we think 50 milliwatts should be the highest for the wearable. But for the since LED far away from that, then it can be a little higher. I mean, the irradiance can be a little higher. For the first thing, we did test the temperature is roughly 40 degrees. When the irradiance is roughly 50 milliwatt square centimeter. Is the answer? Yeah, thank you.
Hello, can I talk?
Go ahead, Hanley.
Yeah. Okay. Really nice work. I think this is answering a lot of questions, and I'll study more of your results. And then James is here, and basically I'll get a chance to present our recent work, basically using both 660 nanometer and 850 nanometer of his machine, and that shows really improvement of reaction time. And it has been a question to me really about 660 nanometer wavelengths, because from my own research experience in the tissue optics area, basically we believe, I mean, this is the FNIRS field that I have been in for the last almost 30 years, starting from Britton Chance's work. And 660, you can see from functional NIRS, we don't really use 660 because light is highly absorbed by blood. But it was a wonder for me for years to say whether 660 can work or not. We know 850 or 800 up to 1070 or 1064 nanometers, they all show some kind of a improvement for cognition. So your work relatively really very much comprehensive and answered those kind of questions. So I guess, again, I'll have more chances to discuss with you. But now one particular question I have now is about Bufferoni correction. Now, again, Luyao, is that your first name? So,
yeah.
So basically, you mentioned that you have conducted a lot of Bufferoni comparison for your EEG data. But then when you present your EEG power, you really did not compare different channels. You were just calculating the total power or the average power across all the channels, correct?
Yeah.
Now, if that's the case, why do you need Bufferoni correction? Because Bufferoni correction is for multi-variable comparison.
No, the Bufferoni is about the the baseline and the durational period, the baseline and the rest time period, and the durational period and rest time period. I mean, here there are three. Yeah.
Oh, so that's more like a ANOVA rather than Bufferoni correct. I mean, that's part of my question. I assume later you may be able to do channel-wise analysis, so then see which part of the brain will have more.
That's a valuable advice. Yeah.
And another quick question or comment is that the way you did for the design is really to check whether they are more alert or not about sleepiness, right? So yeah. How did you control their sleepiness level? Because if I didn't sleep well last night, I'll get sleepier today. But if someone has a pretty good sleep time last night, then they will be more alert. So how do you do that control comparison?
Yeah, I got you. In my study, they were asked to give their sleepiness level by CAS score before the tPBM and after the tPBM. And we have 8 subjects in each group. And as I show in my slide, the differences of the sleepiness before the tPBM, namely the baseline, maybe has no significant differences between the groups. So we assume that it is acceptable, and we mainly focused on the changes within each subject. I mean, the decreasement of CAS scores before and after the tPBM. Yeah.
Thank you. And for your in vitro study, what cells did you use?
Neuroblastomal cells, SHSY5Y.
Okay.
Yeah.
Thank you.
Yeah,
I also
thank
had
you.
a related question about the use of 660 light. I mean, I believe it was when you presented the delayed match to sample task, and you showed that with pulse light, both the 660 and the 850 actually
Yeah.
improved the performance. Now, 660 might have actually seemed even better. So that's kind of, I understand there wasn't a direct comparison in between the two wavelengths. I think this is counterintuitive given the low penetration of red light. And so I wondered, what are your thoughts about that?
Yeah, in fact, I designed 850 nanometers because I'm afraid that 660 nanometers might not brought about significant changes since the absorption and so on. But it turned out the results showed that the 660 nanometers had achieved even slightly better outcome compared with 850. And I looked up some literatures previously. I thought I read some work from previous studies show that 600 to 700 nanometers, this range achieved a good intervention outcomes. But at this time, I cannot explain why 660 nanometer have a slightly better outcome than 850. As I think 1064 like nanometers has been proved its beneficial effects, but LED cannot achieve the wavelengths that long. It's only in the red to near infrared range. So we did not cover that 1064 nanometers like this. We only conducted two wavelengths.
Thank you. Nice work. I think there's time for maybe one or two very quick questions.
Margaret, you're muted.
Oh, I got off. Yes, thank you so much. Can you hear me now? Okay, I had only one comment. It's a wonderful talk from both of you. I mean, you want to think about, I know with the red, there are emissary veins, and we talked about them in our first paper with Anita Saltmarsh way back in 2011. We learned all about emissary veins from Mary Dyson, and these veins have no valves, and they are located near the surface, especially in mid-sagittal, but also in the forehead and many places all around. And those veins protrude outside the skull, and they can take in the photons, and they go, and these veins extend down through the dura mater all the way to the arachnoid and pia. So it's possible you're really helping with vasodilation in some way, especially with your red light emitting diodes. And Joe DeDuro has, I think their whole head red LEDs only that he does work with. He might want to comment on that. Are you, for example, Joe, do you think that you're delivering your red photons to the emissary veins in some way when you work primarily with red?
Well, our head wrap has 4 separate wavelengths. And so the red is only 1/3 of it. We do that to get the scale of the photoreceptors. And so we have 2 different areas. On the surface of the skin, it could just be the blood that's coming in. The emissary veins, yeah, well, there's still plasma and stuff in there that can be, irritated. But we're thinking that we can probably get far enough to get into the meningeal lymphatic vessels that are just under there, which are, yes, you're going to get the veins. We just don't see a ton of the therapeutics from hitting a vein. So we're kind of hoping that we can get in a little deeper to address these the brain's lymphatic system, which are exactly kind of same where you described, along here, around the top, down around the back. And those can happen if you can get the light, maybe, increase a little bit of the circulation there, vasodilation of the meningeal lymphatics. So we're getting a push and a pull at the same time. So we use 4 different wavelengths.
Oh, okay. Then what are your near infrared wavelengths? Are they 850?
We
And
do 635, 810, 904, 1064.
oh, okay. So you go all the way up to 1064.
And it has it's continuous wave. So this is very, exciting to see. I think that there's much to be, investigated on, 8 cohorts. That's a lot of statements. But we're thinking about, using continuous wave just to increase the juice that's going in there.
Okay. Thank you. Anyway, I just want to bring up emissary veins so people understand.
Thank you. Thank you
It seems
both.
like it's a long way to get in there, but you can actually deliver photons to the scalp surface, actually. So anyway, thank you.
Thank you, Marnie, and thank you, Luyao and Muqing. I mean, it's been a very intriguing presentation, and we'll have to think more about this. Thank you all for coming, and to be continued. Take care.
Thank you.
Take care.
Thank you very
Bye.
much.
Bye now.
Thank you.